Hello everyone, my name is Enrico Piperno, and I wanted to make this video to discuss this paper which I found very interesting. The paper is Room Temperature Ultrafast Nonlinear Spectroscopy of a Single Molecule. The reason why I found this paper so interesting is because it really pushes the limits of what we can do in both space and time. The key problem statement behind this paper is how does a single molecule's excited state evolve on femtosecond timescale? A single molecule is on the nanometer scale, and we're interested in the dynamics at the femtosecond scale, so 10 to the minus 15 seconds. The single molecule of interest here is dibenzotyrrolene, and how we approach this problem is by transient spectroscopy. The big picture idea behind transient spectroscopy is by using lasers to first pump the sample to an excited state, and then at some fixed time delay delta t, send a second pulse that probes the sample. Transient spectroscopy looks like this figure I shared here on the right, and we'll be leading towards replicating this plot in my discussion. So light-matter interactions span many fields, from vision, where in just 200 femtoseconds after absorbing a photon, our molecules in our retina changes in response to that photon. Similarly, in plants and bacteria, photosynthesis is nearly perfectly quantum efficient, meaning 99% of photons get absorbed and converted into a useful output, and understanding some of these processes might help us design better solar cells. The question that unites these are what happens to a single molecule after it absorbs a photon? And the answer spans many orders of magnitude, in fact 15 orders of magnitude, from electron motion at the femtosecond scale to protein folding at the millisecond and second time scales. So as a review, I wanted to introduce the Jablonski diagram. So this is a manner to introduce the different energy levels found in molecules. So in the bottom here, we have the ground state, and then I depict in S1 the first excited state, S2 the second excited state, and I also introduce a triplet state. The key point to note here is that the vertical axis is energy. So as we move up in energy levels, we move up in energy. So in S1, we can absorb a photon from S0 to S1. That molecule, that electron now resides in S1, that can vibrationally relax down to the vibrational ground state of the first excited state. And then at some time scales associated with nanoseconds, we fluoresce and we come back down to S0. And of course, the absorption and fluorescence happens at different wavelengths. This vibrational relaxation is on the femtosecond timescale, and this is what we're after in this paper. So in order to measure these processes, you can measure spectra. So spectra measures the amount of light as a function of the wavelength. So here in red, I'm plotting, drawing the absorption spectrum of this molecule. We find this first peak responds to S0 to S1, and the second peak looks like S0 to S2. We can also draw the emission spectra, which I draw here in green, from S1 to S0. And what we're particularly after in this paper is how do these states evolve in time? And the answer, like I mentioned, is transient spectroscopy. So to lead up to the paper, I need to first explain two orthogonal techniques. The first technique is transient absorption spectroscopy, and the second, which we'll see soon, is Fourier transform spectroscopy. So transient absorption spectroscopy looks at the change in absorption as a function of time, and it changes in the absorption spectrum. So the way we do this is we first measure the ground state absorption by sending a single pulse of light composed of many wavelengths. That's why I drew it as a rainbow here. We send this through the sample, and we measure the absorption as a function of wavelength. So we recover that single peak, like I mentioned, that corresponds to S0 to S1. Now, if we first pump the sample, so we bring it to that first excited state to S1, and at some later time send a probe, we have, and we recover the absorption spectrum, we recover these more interesting features. So once again, maybe part of the molecule is not in S1, so we'll recover that same peak S0 to S1. But if the molecule now is in the first excited state, Then we can recover this dark green spectrum, which corresponds to stimulated emission or depleting the fluorescence, or it could also correspond to reaching even higher energy levels from S1 to S2 or to S3. And basically, if we take the sum of these curves and subtract it by this ground state absorption, we recover this blue curve, which I drew here. And if we measure this as a function of time by changing this delay time delta t, we can see the change in this curve as a function of time. That is transient spectroscopy. So the way this is practically done is, like I mentioned, is using pulse lasers. So we'll send a scheme of a probe followed by a scheme of a probe, a pump followed by a probe. And so the spacing between the probe and the pump probe is inversely proportional to the repetition rate of the laser. Yet this delay between the pump and the probe is very precisely defined, and this is usually on the femtosecond timescales. The second technique which we need to understand is Fourier transform spectroscopy. So in normal dispersive spectroscopy, right, we have a white light source, which we separate into its various wavelengths, and then we measure the intensity as a function of wavelength. This is the absorption spectrum if you just flip it on the x-axis. The way this could be practically done is using diffraction grating to separate the wavelengths in space and then shine the light through a pinhole. And we'll rotate this diffraction grating to separate the different wavelengths. Now, in Fourier transform spectroscopy, we have the same white light source and the same sample and detector, but inside the box slightly differently. So now we're actually using interferometry. So this is a Michelson-Morley interferometer, and we have the light is actually interfering with itself once it reconciles from this beam splitter here. As we modulate the moving mirror with respect to the fixed mirror, we retrieve an interferogram. And so if we remove the sample and just measure the intensity as a function of the mirror position, we have this fringe pattern. This bright central peak corresponds to the distance of the mirror that is equidistant from the beam splitter to the fixed mirror, which is perfectly constructive interfering with the light. Yet then we have destructive interference and so on and so forth. Now, if we reintroduce the sample and recover this intensity as a function of mirror position, we have these higher frequency components that reside in our signal. And it turns out that once you Fourier transform this signal and divide it by the background, that is without the sample, we recover this intensity absorption spectrum. Now, the reason why spectroscopy is so hard to do with single molecules is because they're so small. So because of the diffraction, the wave-like behavior of light and the diffraction limit, if we tied a laser to its smallest focal spot, the smallest we can get is this area S. Yet single molecules are at this order of magnitude in centimeters squared. This is the so-called molecule cross-section, sigma. If we compute the probability of absorption of a photon per molecule, we find that it is the ratio of these two areas. And this is extremely small, so 10 to the minus 6, or 1 in a million photons, actually gets absorbed by the molecule. And so the key is to measure the fluorescence of the molecule. So as we mentioned, fluorescence happens at a different wavelength, and it happens at much later time scales in absorption. And actually, by both the wavelength and the time, you can remove the absorption signal and only look at the fluorescence signal. You can filter the absorption signal, I should say. So now speaking about the paper, the single molecule, the sample of interest is dibenzotyrrolene. So this is a molecule chosen out of convenience. This is because it is has a high quantum yield and it is very photostable, meaning that it fluoresces for very long time scales before being photobleached, and it also absorbs and emits at wavelengths which are sensitive to modern day detectors and cameras. To further simplify the sample for this experiment, they actually lock the molecule in an anthracene crystal, as shown here. And this really locks the molecule in place in space, and because it absorbs and emits at different wavelengths, where this does not show up in our signal. It is not even in the background. So the key concept behind this paper is to measure femtosecond dynamics. by measuring fluorescence, which happens at nanosecond timescales after. So when reading this paper, this was really the trickiest part for me to understand, but I think I can explain a little bit better now. So once again, here I'm measuring and drawing the Jablonski diagram for a single molecule, except now we have these potential energy wells. So when our molecule is in the ground state, it has this wave function, which is the wave function for the electrons in the molecule. This can be pumped to that first excited state of the molecule. Now, that wave function has its own dynamics. It can vibration relax, like I mentioned, go down to that ground vibrational energy level, but it can also oscillate and so on and so forth. The pump is bringing this electron from the ground state to the first excited state. Now, the probe arrives at a very specific delay time, delta t, and this depletes the fluorescence from that molecule. And exactly how much it depletes depends on the excited state spectrum at that exact moment. So while we measure the fluorescence at nanosecond timescales later, the intensity of that fluorescence really is a function of those femtosecond dynamics because we depleted the fluorescence at those timescales. So this is a plot in the figure in the paper that they provided. So once again, we're pumping the molecule, and then we're sending a probe at some time delay, delta t. Then we recover the fluorescence much later time, but because of that probe, we can actually use the fluorescence and measure that intensity as a function of this delta t and recover that excited state spectrum. So as a further complication, we use the Fourier transform spectroscopy method that I mentioned. So not only are we pumping a sample and then probing it, our probe encodes different frequencies in it. And then when we take the Fourier transform of that signal, of the fluorescence, we recover that excited state spectrum. So once again, this is showing the fluorescence depletion of that event. So at negative time delays, the probe is actually reaching before the pump. So the probe is actually now exciting the molecule to that first excited state, and the pump is depleting the fluorescence. But you can also see that for positive time delays, where the pump comes before the probe, we see that same depletion of fluorescence. Now, this is what I was mentioning about encoding different frequencies. So we're actually shaping the pulse of the probe in different frequencies and what they call effective delays. So this effective delays you can think about as two identical pulses constructively and destructively interfering. And if I actually plot what those look like, you would see these plots that would look something like this. So this effective delay is completely distinct than this time delay delta t, which we're actually using to recover that fluorescence, that excited state spectrum. But as we modulate the effective delay, as we shape these probe pulses at fixed delta t, so these values here are at those fixed delta t's, we reconstruct the interferogram. So this is like I showed for the Fourier transform spectroscopy. Now, if you Fourier transform these, we actually recover the excited state spectrum. So once again, each line here is for a fixed time delay between the pump and the probe. And the way we recover this is by Fourier transforming the interferograms, this signal, which we recovered by modulating the probe shape. So for every line here, we're shaping the probe in many different ways, Fourier transforming, recovering that spectrum. Hopefully that's clear. The way this is practically done is through very fast laser. So this is the optical diagram, which I kind of tried to draw from what the authors mentioned. So from what I understand, we have the laser source, followed by a pulse shaping scheme, followed by a scheme to create those probes and pumps and delay them, followed by our detection. So we're using a Ti-Sapphire laser, very fast 6 femtosecond pulses in the red and IR region with a 85 megahertz repetition rate. Immediately after, we have some passive pieces of optics that are correcting for any dispersions to our pulse lengths, which might happen downstream in our optics. So once we correct the dispersions for our pulses, we then try to shape these pulses. So here we're using a 4F diffraction grading scheme. What you need to know here is that we're using a spatial light modulator at the Fourier plane of this light, meaning that space now encodes frequencies. So the spatial light modulator you can think of as a grid of many pixels. With each pixel you can dim as you want. You can program this spatial light modulator. And so here we're actually programming the pulse to carry very distinct frequencies because we're in the Fourier plane of that light. And the frequencies that we choose, or that the authors choose, are these frequencies for the probe and these frequencies for the pump. And then we're going to be detecting our fluorescence here in the gray region. These probes and pumps are going to be about 25 femtosecond long, and that's the temporal resolution of our system. So once we take those Ti-Sapphire pulses and modulate them to contain these frequencies, then we want to be able to separate the probe and the pump and to move and delay these with respect to each other. So now we're going to be using a 50-50 beam splitter, or sorry, a dichroic beam splitter to separate the pump colors to the probe colors. And then we're going to have reflecting mirrors that we can move back and forth to optically delay the pulses. So for 10 femtoseconds, if we wanted to achieve that delay, we'd have to move the mirror 3 micron, or I think 3 micron divided by 2. Now, if we want to detect our sample, we excite our sample with the pump probe and sending our probe pulse at a very fixed time delay later. This is going to be focused onto our sample using a reflection objective, and then we recover the fluorescence with an objective, fluorescence filters, and our electron multiplying CCD. So we've seen this figure many times before in this presentation. Now, the actual intuition, the reasoning the authors provide to explain the spectrum is here. So at very early time delays, we find that this spectra is wide, it's kind of flat. But at later time delays, we find these two peaks in our spectra. And they explain these by vibrational relaxation. So you can think that if we excite our molecule and immediately recover that excited state spectrum, this molecule has many different avenues of fluorescing because it could be in all these different vibrational energy levels and it could decay to these other vibrational energy levels. So each of these corresponds to different frequencies. As you add them up, you get this sort of wide spectrum. Now, if we wait some femtosecond later, the electrons that actually arise in this higher vibrational energy level can decay down to the ground vibrational energy level of that first excited state. Now that electrons have less avenues to fluoresce, and so they correspond to maybe two wavelengths, which correspond to those two peaks. So this is the author's explanation for this excited state spectra plot. So some discussion and drawbacks. So for this paper, we're only probing the first excited state. That's okay, but we kind of lose information of what could happen in the second, third, or triplet states. Also, our sample is highly idealized. So it is very highly photostable, meaning it emits billions of photons before photobleaching, and are also embedded, locked into a crystal. In real biological samples, in a wet environment, this won't be the case. And usually fluorescent proteins are not nearly as photostable as dibenzotoluene. Similarly, I wasn't able to recover any signal to noise ratio, although they mentioned that the background signal is the signal they recovered outside of the spectra that they were interested in, so this gray region, but it wasn't clear to me what these values were and thus what their SNR was. So these are the papers I used to review this paper. So I want to finish here by speaking and explaining some of the applications of this technique. The first is in actually probing plants' extreme quantum efficiency. So in this diagram here, this is actually a chloroplast of a plant or bacteria, where photons gets absorbed by individual chlorophyll molecules, which creates these excitons, which you can think of as small little voltages, which are created by electron-electron hole pairs. This exciton then needs to travel to the photosynthesis reaction center, where it can be converted into useful output like the sugars and oxygen that we breathe. However, each step that takes from a single chlorophyll molecule to that reaction center loses heat, and so it is in the plant's best interest to find the most efficient path, or the shortest path, from where the chlorophyll originally absorbs the photon to the reaction center. And it is theorized that the way the plant actually finds the shortest path is through many quantum principles, such as quantum entanglement and quantum coherence, where in essence, the exciton explores many paths at once in parallel. So it entangles all these states, and then when it finds the most efficient path, it collapses and actually converts that energy to useful output. So the way this was measured is through transient spectroscopy. So once again, we have the spectrum, and we measure the spectrum at very fast timescales, femtosecond timescales. This beat that we could find here in our spectrum, this is known as the quantum beat, and this is a signature of that quantum coherence in photosynthesis. The second application is how do we design better solar cells? So a fundamental limit is the Shockley-Queisser limit, which states that 37% of the solar energy can be converted to electrical energy at maximum efficiency. This is because for single layer silicon solar cells, we can only extract one exciton for every photon. However, we can, through a process known as Singlet fission, actually produce 2 photons for the price as 1. So now, once the molecule absorbs a photon, it is excited to that first excited state S1. Now, if we bring another photon very close together, we find that this electron in the singlet excited state can be converted into 2 electrons into 2 triplet states. Then when these decay, they phosphoresce, and they actually emit those 2 photons, which we can then use to beat this fundamental limit in solar cells. And once again, the way this was measured is through transient spectroscopy. So we find here the spectrum as a function of time, and we can see changes in the spectra are actually a signature of what is occurring in singlet fission. Thank you very much for listening. I hope you learned something, and I'll link the papers and time points of this talk in the description.